Okay. Play. Yes. Okay. Yes. All right, we're in business. Okay, so this section is going to go over how gene expression affects development. The control of gene expression plays an important role in the growth of eukaryotes as different cells become specialized to perform different tasks. This is super important because the entire point of gene expression is to control which genes are expressed, meaning which proteins are made and when. So if you think about all of the hundreds of different types of cells in your body, they are made differently because they have different functions in your body. All of that is completely based on gene expression. So for example, in a red blood cell, you're going to have the genes turned on that code for the protein hemoglobin, because the purpose of a red blood cell is to transport oxygen, and the protein that transports the oxygen is hemoglobin. Versus, let's say, a skin cell, which doesn't have anything to do with transporting oxygen, will not need to even make hemoglobin ever, because it doesn't have to transport hemoglobin. So at different points of development, you're going to have different genes being turned on and off, meaning that they are expressed, meaning either expressed being turned on or not expressed being turned off, which means you're going to be making different proteins at different points of development. When the expression of genes is altered by mutations, for example, abnormalities and even cancer can result. Notice I don't say does result or will result, can result. Please keep in mind big picture, which is the most difficult thing to do when you're studying biology. When you look at like all of these very specific kind of not procedures, but processes, it's easy to get lost in the details. What do they say? You can't see the forest for the trees. So if you're in the middle of the forest, all you see is tree, but you don't see the forest. I want you to be conscious of looking at the forest. So think about during development, from the fusion of egg and sperm, how many different changes occur as the embryo grows into a fetus and then finally as an organism. All of those changes are a result of genes being turned on and off during different times of development. So not only that, but also understanding that like 80% of our DNA just is unused most of the time. Thinking about the fact that 99.8%, let me say that again, 99.8% of our human DNA is genetically identical, meaning exactly the same as the DNA molecule of a chimpanzee. So you're talking about 0.2% of our DNA, which separates us from our closest living relatives, which are the chimpanzees. So if a mutation happens, it depends on where it happens. It might happen in a part of DNA that's never expressed, so there's absolutely no effect whatsoever. It might occur in a vital protein that's necessary for the development of a fetus, which means the fetus is not going to develop properly, which could result in something called a miscarriage. So if a woman is pregnant and during development, there is a mutation in the development or in gene expression, the fetus could be spontaneously aborted, which means it like the female's body like pushes it out because it's not normal and it can't result in a viable organism. So we're going to go through all that. Oops, that got cut off. That should be cell differentiation. So what you see up there in the top corner, oops, can I go backwards? Okay. What you see in the top corner, this is day 1 after fertilization. You see day 2 in the morning, day 2 in the evening, day 3, day 4, day 5, and day 6. This is after an egg is fertilized by sperm. You can see that it's gone through one mitotic division. So it's gone through mitosis one time. So one cell becomes two. That evening, two cells become four. So there's one more version of mitosis. Remember when I told you way back when that eukaryotic cell division takes a long time? It's like 24 hours for one cell to divide two times, but prokaryotes divide like every 20 minutes. This is what I'm talking about. So between the morning and the evening, you have only one round of mitosis. Day 3, again, you've got one more, so 4 becomes 8. But also notice it doesn't change size. So as the cells divide, the cells are becoming smaller. Finally, what you have here is something called a morula. We're going to talk about this more next year when we go into human anatomy. A morula is basically like, I want you to think about a soccer ball. So the cells are around the outside, but the inside's filled with fluid. This is how we all start, right? 6 days after fertilization. Just a little ball, a hollow ball of cells. All multicellular organisms begin this way as something called a zygote, okay? A zygote is a fertilized egg. Every cell has the same genes, but only a fraction of them are expressed. Genes are going to be turned on and off as different proteins are needed during development. There are very specific proteins and enzymes that are made during this process of development that are not needed once you are born. And so that's why gene expression is so important. Cell differentiation is exactly what it's describing. It's a process, of cells becoming different. The development of stem cells, which are what you saw in the previous picture, stem cells have the potential to become muscle cells or blood cells, nerve cells, cardiac cells, etc. We have several hundred different types of cells, not 700 different cells, 700 different, not seven, several hundred different types of cells in our body. Our body has over 30 trillion actual cells, 30,000, million, billion, trillion. 30 trillion cells. There are several 100,000 cells that are being destroyed like every day and being replaced. Muscle cells function in movement. Liver cells make enzymes to break what? Break bile, I think. Break down fat. Oh yeah, bile. That's the same as bile. Sorry, I got cut off. As an organism grows, the organs and tissues develop to make a characteristic form. This process is called morphogenesis. I know you guys study Christology, so you know what, well, maybe you know what Genesis is. This is the first book of the Bible. It means to create, and morpho means shape. And cis, of course, is process. So gene is create. So the process of creating the shape. This is human development in 36 weeks. Should be about 36 weeks. I can't see because this stupid bar is covering the bottom. Hold on. Let me move that to the top. There we go. All right. This is up to 24 weeks. So I don't know about here in Thailand, but under American law, at 24 weeks, so week 24, that's it's called a viable fetus, which means it can survive on its own. This entire time, as an embryo and then a fetus, it cannot survive outside of the mother. Legally, right? Legally. Just want to make that clear. I'm not here to talk about morality at all or ethics. Not my job.
In the animal kingdom, just about wherever we find brains, they're wrapped up in heads. But being so important, you think evolution would have, I don't know, looked after the brain a bit better than just this thin candy shell. But when you think about it, there's a lot about the human body that doesn't immediately make sense. We breathe through the same hole that we eat with. We grow more teeth than we can fit in our mouths, and we built our amusement park next to the garbage dump. To craft this brain story, we need to head back to the beginning, and we're gonna need some help.
From Braincraft.
The very first animals didn't have brains or heads or really much of anything going on, but our ancestors did learn left from right.
Around 550 million years ago, the first animals with bilateral symmetry branched off from those with radial symmetry. A radial animal, like a jellyfish, only has two body axes to play with, from head to tentacles and out from the middle. They're a bit two-dimensional.
Bilateral animals suddenly had a new dimension to play with. Not only could they have a top and a bottom and grow out from the center, they could have a front and a rear. So many new possibilities. Every bilateral animal uses the same set of genes to control how its body is organized head to tail.
This is one of the most amazing things that I found in biology is what he's going to talk about now, the Homeobox. I'm going to get to it in a second, but I want to just rewind for a second, pay really close attention to what he says. He said every single animal has exactly the same blueprint for the body, meaning where the head goes, where the eyes go, all of that. It's absolutely fascinating.
Every bilateral animal uses the same set of genes to control how its body is organized head to tail. These genetic choreographers, called Hox genes, control the developmental dance performed by hundreds of other genes, instructing an embryo to put the head here or tail there. Hox genes themselves are clustered on the chromosome, strung together in the same order as the parts of the body that they organize. This is the basic body plan that nature has used to build everything from nematodes to regular toads, from water bears to polar bears, and humans to honeybees. The Hox genes of complex animals like humans or mice aren't so much different from, say, a fly. We just have more of them. Along our evolutionary journey, these architects were duplicated, allowing the redundant copies to specialize and sculpt new forms.
Oof, I wanted to catch that right at the end. So look at that. Wait, where'd it go? No, there. So between a fly, a mouse, and a human, look at how this all measures up. They're all in exactly the same place. So like this is where the head goes. You've got like a neck area, whatever. All of them is exactly the same. The only difference is the more, let's say, complex the organism is, say for like a human, you just add more parts. It is so freaking weird. We're all built exactly the same way. I just want to clarify that we're talking about bilateral animals. Bilateral animals, they have a front and a back. They have a left side and a right side, and they have a top and a bottom. So that's different from animals called radial animals, like a starfish, which I can't draw, so I'm just going to do that. Starfish, which has a center, and it can radiate out from the center, and it has a top and a bottom, but it doesn't have a front and a back. There's no head and tail there. We'll get into all of that.
New forms. Those early animals upgraded from random jumbles of just a few nerves into telegraph lines from tip to tail, but they still had no central nervous system. No brains, no elaborate spinal cords, just the simplest of wires that connected front to back. Turns out that in order to spell brain, you need an I.
If you're in the business of eating, it helps to have them on the end of your body where the food goes in. The other end is just not pleasant. So animals focus their eye spots and other sense organs on their eating ends, and so more and more nerves began to cluster nearby. This marks the beginnings of a brain.
We know that eyes evolved more than once in different branches of life. On those branches, the bodies and nervous systems continue to grow more and more complex. And as those early brains became more powerful and valuable, one branch of animals started to protect their main nerve cord. Later, as sensory organs like eyes and scent receptors became increasingly powerful, so did the brains that process their information. They were worth protecting. Well, yet another group of animals called the craniates began to wrap theirs in protective skulls. Now, hardened skulls meant deadlier jaws too, so some animals developed fins, all the better to escape with. Now, this arms race led to more and more bony body parts. Our ancestors must have been Daft Punk fans.
Harder, better, faster,
The first vertebrates that walked out of the water would diverge into animals as diverse as dinosaurs and dogs, but their basic body plan was set. Brain inside the skull, up at the eating end, close to the sensory organs. Evolution's kind of like Taco Bell, an infinite number of variations from the same few ingredients.
Great. Now I
That's
want
how
tacos.
evolution planted the seed that put your brain in your head. Now, why don't you go put it to good use?
And head over to Braincraft to find out the amazing way the human brain grows. Stay curious.
Hey, that's my line. This video is part of a
Okay, so first we're going to talk about homeotic genes. These are regulatory genes that determine where certain body structures will develop during morphogenesis. So going back to this picture here, where we start from, oops, wanted the laser. Where we start from, like, why is it that our head develops here and not like at our butt instead? Why do we have 2 arms instead of 4 or 8 like a spider? All of those are in homeotic genes. So again, you can see the color matches here where the body part is. So if you follow the colors, you can see that it literally follows down the length of their body from head to tail. Okay, so the word development got cut off there. They're the master genes of development. It's kind of like a blueprint, if you will, of where the body parts go.
So what do we learn by looking at 600 million years of animal history? Evolution's tinkering with mammalness to make whales. In the same way, it's tinkering with fishiness to make tetrapods. And it's tinkering with animalness to make all the different body plans that we see. All these different creatures are variations of the same theme, restated over and over again. The question was, what was evolution tinkering with? One of the remarkable discoveries of the last 20 years is that evolution's not tinkering with the bodies. It's tinkering with the recipe, the machinery that builds bodies. What is that recipe? What is that machinery? It's the genes.
Scientists had long suspected that embryos held clues to how animals evolve. All embryos start out as clusters of nearly identical cells. But soon, an embryo partitions itself into specialized segments, which develop into the final form of the animal. What controlled this process? How did the embryos know what shape to take? One researcher, Dr. Ed Lewis of Caltech, studied this question for 30 years by crossbreeding thousands of flies. Lewis's work led him to a controversial idea. He proposed that a surprisingly simple mechanism was shaping embryos. He wrote that each segment of the fly was being directed to grow by a single gene. A small set of genes, a kind of genetic toolkit, appeared to be laying out the entire body. In 1994, Walter Gehring of the University of Basel isolated the gene that triggered the growth of eyes in fruit flies. The gene was called eyeless because flies without it developed with no eyes. Gehring knew of a gene in mice that worked in the same way. He wondered, were the two genes the same?
And this question
Okay, this is one of the most amazing things I've ever seen. And I want to stop here and make sure that everyone's paying attention because this is going to blow your face off. So there's these flies that can be, I don't know if they're born, but hatched, I guess, or develop without eyes. And this scientist knows that there is a gene in mice that does the same thing about like the eye development, but I want you to understand that insects have completely different eyes than mice do. Mice have a simple eye, which is the same thing we have. One lens, one eyeball. Insects have what are called compound eyes, and each, I guess, what you call an eyeball has thousands of lenses. It's a completely different eye structure. Now, I want you to please pay attention to his results. They're absolutely amazing.
Tested by taking the mouse gene and putting it into fruit flies to see whether flies can understand the message of the mouse.
Gehring replaced a fly's gene for eyes with the mouse gene.
And to everybody's surprise, the mouse gene works perfectly well and can induce a compound eye in the fruit fly.
I mean, how freaking nuts is that? They took the gene for a mouse eyeball and stuck it into the embryo of a freaking fruit fly. Not only did the eyeball develop in the correct place with the correct number of eyes, but it also developed a compound eye, which mice don't have. That is like head blowing up emoji.
The fruit fly grew normal fruit fly eyes using a gene from a mouse. Not only did the two creatures use the same mechanism, they used the same gene.
So what this means is in some ways, some sense, evolution is a simpler process than we first thought. When you think about all of the diversity of forms out there, we first believed that this would involve all sorts of novel creations, starting from scratch, again and again and again. We now understand that no, that evolution works with packets of information and uses them in new and different ways and new and different combinations without necessarily having to invent anything fundamentally new, but new combinations.
Suddenly, the commonality of form among animals was understood. Animals resembled each other because they all used the same set of genes to build their bodies. A set of genes inherited from a common ancestor that lived long ago.
And there's only one inescapable conclusion you can draw from that, which is if all of these branches have these genes, then you have to go to the base of that, which is the last common ancestor of all animals, and you deduce it must have had these genes. So the whole radiation of animals, the whole spring of animal diversity has been fed by essentially the same set of genes.
So cool. So here we have a fruit fly versus a human, and you just look at how they are similar. Basically, the body parts are forming in the same pattern, which is what they kept saying in the video. We're all basically the same, or like different versions of the same thing. So when a homeotic gene is expressed, regulatory proteins are formed. These proteins then regulate development by switching the developmental genes on or off. This is going to increase or decrease rates of cell division in various areas of, I'm guessing, developing body or something like that. So, for example, let's just take a person, right? Now, during development, we start as like a blob thing, right? Like a ball of cells. And then I'm just going to try to like simplify this as much as possible. At some point, there are cells in a certain area that are like, okay, well, the homeotic genes say an arm goes here and an arm goes here. So they're going to turn on gene expression so that cell division is increased in this area and that area. So you have two appendages, two things that are coming out from the side. Same like at the head area. Oh, okay, well, we need a neck here and we need a head here. So that's why genes get turned on and off. Same thing with legs. You have extensions that need to come out from the body. If you're an insect, then you have a body, and then you're going to have that 6 times. Is that 5? What did we go? 5, 6. I know my art skills are just amazing. but insects have 6 legs. So they're going to have 6 legs coming out from the bottom. We have 2 arms coming up from the top and 2 legs coming out from the bottom. This is all variations of the same thing. Now, this is an insect that is near and dear to my heart. I have killed many of these creatures in my genetics lab, which is why every time they land in my drink, I try to save them now. It's a species called Drosophila. The whole name is Drosophila melanogaster. Drosophila melanogaster. I used to be able to tell which ones were male and female. I don't remember anymore. But this was the class that was taught by the professor who knew both Watson and Crick. Fascinating. But we killed a lot of these guys. So each homeotic gene shares a common sequence of DNA of about 180 nucleotide pairs. These are called the homeobox. These are specific DNA sequences in the homeotic genes that regulate development. So these are basically going to tell where specific body parts go. So if I go back here, each of these is a homeobox. Okay, but together they're called homeotic genes. Oops. Okay, so this is obviously a mutation in a homeobox. Here you have a normal fly head. This is what I mean by compound eye. You see every single dot here. Each of those dots is a lens, and each lens is going to have an electric signal to the brain, which will form its own image. So if you imagine what an insect vision might look like, if you've ever looked through a kaleidoscope, if you don't know what those are like the tubes where at one end of the tube you put like little pieces of colored things, and then you've got mirrors in there. So then when you turn it around, it forms this kind of like psychedelic kind of image. It's kind of like that. So normally you don't have legs sticking out from your head, right? But this is a mutation. You can see appendages here that are sticking out from the head in this fruit fly. That is when you have a mutation in a homeobox, where there's the part, the homeobox, which deals with the head, and there's a mutation, so legs grow there. Oops. Why? Okay, this is another mutation here you can see in the HOX, which HOX is short for homeobox, HOX. Gene D13. So we've been able to identify our HOX genes, and you can see it's called synpolydactyly. So poly means many. Dact is something to do with fingers and toes. So you can see, I'm not really sure what the prefix syn means, and I don't have my scientific root word sheet here at home. But obviously you see where there's either more than one finger where there should be one finger. Same here. You get 1, 2, 3, 4, 5, 6 fingers here. Or you have multiple fingers that are fused into one. Synpolydactyly, yeah. Trying to think what syn would mean. I don't know. All right. What time I have? Ooh, 9:40. Oh gosh, that just flew by. All right. I'm not going to get into oncogenes and cancer yet. I'll save that for next week.